Music licensing reimagined. Artlist.io. 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 Artlist.io. Music licensing reimagined. Artlist.io. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Artlist.io. Music licensing reimagined. Artlist.io. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Artlist.io. Music licensing reimagined. Artlist.io. Artlist.io. Music licensing reimagined. 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 Artlist.io.
Hello, everyone. Good morning. Please, can everyone hear me? Can you give a thumbs up or maybe comment on our platform if you can hear me? Okay. All right. Good morning, ladies and gentlemen, distinguished guests, speakers, esteemed speakers, honorable participants, both at the University of Nigeria and Nsukka, and joining us online from around the globe. Good morning once again, and then welcome to the 2024 AODI Science Lecture and Workshop. I am Andrews Adiasante, your Master of Ceremony for today's event, and I'll be moderating the event online, but will be joined by my colleague, Chiamaka Indulu, who will facilitate the event on ground and in person. I'm absolutely thrilled to be here with you. Today promises to be an exciting journey through the fascinating world of science communication and medication, and I'm already feeling the buzz of anticipation in the air. First off, let's give a huge round of applause to our host, the University of Nsukka, for welcoming us into their prestigious hall for the second year running. Your support in advancing scientific education and research is nothing short remarkable. Now, how about another round of applause to our fantastic founder, the American Society of Biochemistry and Molecular Biology. Their generous support has made this event possible, and we are deeply grateful for their unwavering commitment to scientific excellence and equitable education. We are incredibly fortunate to have an array of distinguished speakers with us today. From the hallowed hall of the University of Cambridge and Oxford to our very own University of Nigeria in Nsukka. These experts have generously taken time out of their busy schedules to share their knowledge and inspire us all. Your thoughts will undoubtedly spark innovation and enrich our understanding of science and pharmaceutical innovation. So we say a welcome to our speakers. To everyone tuning in online and present at the Princess Alexandra Auditorium, UNN, a big shout out to you. Thank you for joining us both in person and virtually. Your presence and participation are vital, and we are excited to bridge the gap between continents with the power of technology. Today's agenda is packed with enlightening sessions from keynote speakers to hands-on workshop. We will journey through the intricacies of drug development, explore groundbreaking therapeutic approaches, and delve into the science of multi-drug transporters. Plus, we would have some interactive workshop where you get your hands into the molecular modeling and computer-aided design. Sounds exciting, doesn't it? I encourage everyone, whether you are at the auditorium or joining us online, to actively engage in today's session. Ask questions, share your thoughts, and make the most out of this unique opportunity to learn and collaborate. Who knows? You might just meet a future research partner or spark an idea that could shape the future of science and healthcare. Before we dive into today's activity, let's take a moment to appreciate the incredible team at the Africa of Our Dream Education Initiative. Their dedication and hard work behind the scenes have made this event possible. Let's give them a round of applause also. And with that, let's get this show on the road. To kick off, please join me in welcoming Dr. David Izioku, the visionary founder and chairman of the Board of Trustees, for his opening remarks. David, the stage is yours.
Thanks, Andrew. It's really exciting to be here. So I've got some flight challenges, which has made it impossible for me to be on ground with you guys right there in the UNN. But I'm still happy to, give this opening remark on my own part this morning virtually, and then after that, try to make my way down to Nsukka. So I've titled my talk this morning around advancing science communication and looking for a way to bridge research and the real world impact, which is basically what we do at AODI. So it's an honor and privilege to come before you this morning as the founder of this foundation. As we gather for this science communication workshop, I'm always really inspired by the collective vision that unites us, which is just advancing scientific literacy and fostering innovation across Africa. So when I reflect on the journey that AODI has gone through, I'm reminded of the power of education and collaboration. Since our establishment in 2017, when I first arrived at University of Cambridge for my PhD, AODI has been steadfast in our mission to empower youth. So by testament to the belief that access to education can transform lives and ultimately our continent. Through scholarships, grants, mentorship program, we've reached over 5,000 students, providing them with tools and opportunities to succeed. Our global mentorship program continues to be a cornerstone of our efforts, fostering growth and development among Africans' brightest minds. So today's workshop is a continuation of what we started last year when we launched the first science conference and workshop right there in UNN with over 500 participants physically present. And that exemplifies our commitment to nurturing talent and fostering education exchange. So we are here again this year, and this time to explore the fascinating world of science communication, to learn from esteemed experts, and to inspire the next generation of leaders in science and technology. Our theme this year is going to be science communication. Now let's discuss medication. And this underscores the importance of clear and effective communication in the scientific community. Medication play a critical role in healthcare, offering solutions to manage, treat, and prevent a wide array of diseases. Yet, the complexity of drug development, the mechanism of action, and the intricacies of drug-target interactions often remain misunderstood. This workshop aims to therefore bridge that gap, enhancing our understanding and communication of these vital processes. So medication are at the heart of modern medicine, as you might know, representing the culmination of years of research, development, and rigorous testing. The journey of a drug from the laboratory bench to the patient bedside is a testament to human ingenuity and perseverance. It involves multidisciplinary collaboration among chemists, biologists, pharmacologists, and clinicians, each contributing their expertise to create safe and effective treatments. We are privileged today to have an exceptional lineup of speakers who will share their insight and expertise with us today. Starting from Professor David Spring from the University of Cambridge, who will open up with an introduction to drug development, providing a comprehensive view of the drug discovery process. And then later, we are going to move all the way to Professor Franklin, Professor Hendrick, Professor Ikechukwu, all speaking on different, very important topic that is going to be a milestone in our journey to understanding all of this. As well as Professor Angela, who is going to then close the keynote speaking with the drug and target discovery of a unique kind of disease of the Duchenne muscular dystrophy. And so these distinguished speakers will not only share their knowledge, but also inspire us to think critically and creatively about the challenges and opportunities in science communication and drug research. In our ever-evolving world, the role of medications continues to expand, addressing new and emerging health challenges. The development of novel therapeutics for conditions such as cancer, genetic disorder, and infectious diseases highlights the dynamic nature of pharmaceutical research. As scientists and communicators, it is our responsibility to ensure that these advancements are understood and accessible, promoting informed decision-making and public trust in science. Our efforts at AODI have always been about bridging gaps in knowledge, gaps in resources, and as well as in opportunities. In the past, with the support of institutions like the Cambridge Africa Initiative, we enhanced research capabilities at the University of Nigeria, Nsukka, by providing advanced research equipment and high-performance computers. This initiative has significantly improved the research infrastructure, enabling groundbreaking work in chemical and biological science at the university. As we embrace the future, we must acknowledge the transformative power of technology. Tools like artificial intelligence are revolutionizing how we learn and work. In fields such as computational biology and molecular dynamics, machine learning is paving the way for new discoveries and innovations. Our challenge is to ensure that these advancements are accessible and beneficial to all, particularly in emerging countries like Nigeria. Our journey is about building strong institutions and investing in human capital. It is about creating an environment where knowledge flourishes, innovation thrives, and opportunities abound. Universities like the University of Nigeria and Nsukka play a pivotal role in this endeavor. By fostering a culture of research and innovation, these institutions can drive transformative change and shape the future of our nation and continent. At AODI, we are committed to building a better world through education, research, and collaboration. We believe in the power of partnership between academia, industry, and government to bridge the gap between theory and practice. By working together, we can create a sustainable future where every individual has the opportunity to thrive. As we embark on this workshop, let us remain steadfast in our resolve to advance science, communication, and innovation. Let us embrace the spirit of curiosity and collaboration, and let us commit to making a difference in our communities and beyond. In conclusion, I encourage you to seize this opportunity to learn, to engage, and be inspired. The future of science and medicine depends on our collective efforts to communicate, innovate, and drive change. Together, let us build a future we can all be proud of. A future where science serves humanity and every dream has the potential to become a reality. Thank you, and I wish you all a prosperous and enlightening science communication workshop.
Wow, that's a lightning opening or welcome address. Thank you very much, Dr. Izioku, for those inspiring words. We are all set for a day of learning, sharing, and innovation. So up next, quickly, because we are behind schedule, let me welcome Professor Ngwaguma Chima Bennett, who is the head of department for biochemistry, to give his opening remarks. The floor is yours, Professor Ngwama. Okay, I've been informed that Professor Ngwangoma is not around. So up next, we would have Professor Onyinshi. Please forgive me with the pronunciation as I'm not Nigerian. Please let's welcome Professor
I think Professor is probably still not available. I think we can move to Professor David Springer.
Okay, that's great. So I think up next would be my opening remarks. Don't you think I would have to give mine? Or what do you think?
Yeah, I'm just curious of David's time. Maybe we can let him jump onto the call. Maybe after him we can do something. Yeah, thanks.
Okay, great. So up next we would have it's an absolutely pleasure to introduce our distinguished keynote speaker for the day. Professor David Spring's illustrious academic career began with his BA Honours NMA in chemistry from the prestigious University of Oxford under the supervision of the renowned Sir Jack Baldwin. He earned his DPhil focusing on the proposed biosynthesis of manzamin alkaloids, a remarkable start to what has become a profoundly impactful career. Professor Spring's quest for knowledge then took him across the Atlantic to Harvard University, where he served as the Wellcome Trust postdoctoral fellow and a Fulbright scholar, working alongside the eminent Stewart. These experiences laid a solid foundation for his future contributions to the scientific community. In 2001, Professor Spring joined the University of Cambridge, initially as a BBSRC David Phillips Fellowship. His talent and dedication quickly earned him a series of prestigious positions, like a lecturership in 2016, senior lecturership in 2018, readership in 2011, and a professorship in 2013. Throughout his career, Professor David Spring has been decorated with numerous accolades, underscoring his profound influence and expertise in chemistry. His extensive research portfolio keeps volume about his commitment to advancing scientific knowledge and translating that knowledge into the real-world application. Today, we are honored to have Professor Spring with us to present on the topic that sits at the heart of his work, from bench to bedside, the journey of drug development. His insights and experiences are sure to enlighten and inspire us all, providing a deeper understanding of the intricate process of bringing scientific discoveries from the lab to patients. Without further ado, please join me in welcoming Professor David Spring as he takes the stage. Thank you for joining us, Professor.
Thank you, Andrew. Thank you for the introduction, and thank you, David, for the invitation. Can people see the slides on the?
Yes.
Yeah, there we go. So, my title of the talk is From Bench to Bedside, the Journey of Drug Development. And what I will do is give you an overview in this of the drug discovery process, and have a very broad outline of this journey from drug discovery to development to clinical trials and to approved markets. I'm aware that this afternoon you'll have more detailed talks with some specific examples of drug discovery in certain areas. So I won't give you any specific examples in this limited time. If we look at this drug discovery process, you can see that at the top of the slide, you can see the number of compounds at individual stages. So typically, if you were doing a small molecule drug discovery, you would start off with tens of thousands of compounds for screening, and then you'd be identifying a smaller number which you take forward through drug development and preclinical research. Then ideally you would have one or two compounds entering clinical trials, maybe just one compound. This enters the process of clinical trial development, and if all goes well, you can then apply for drug approval, which is then reviewed. This whole process of drug discovery takes 10 to 15 years typically. So, of course, there are exceptions, and the recent COVID pandemic showed some drug approvals could be a lot more rapid in emergency situations. But it's typical for drug discovery to take 10 to 15 years. If we look at the very first stage of drug discovery, and I'm going to go through this in a bit more detail in the second half of the talk, there is the process of identifying your small molecules by multiple testing criteria. And so we'll look at that in detail, along with the drug development and preclinical research that might be going on. This typically takes 3 to 6 years before you will get into a phase 1 clinical trial. The clinical trials themselves take 6 to 7 years, of where there's phase 1, 2, and 3, which would typically be identified. And phase 1 is starting with a limited number of sometimes healthy volunteers, say 20 to 100 people, and that would just be looking at its sort of general dose management of the drug. But phase 2 clinical trials is where a lot more stringent criteria of whether the drug works happens. And this would involve several hundred patients and take longer. And phase 3 takes even more patients to check things work. So all things going well, then this can be then applied to become a new drug for approval. So let me now just look at some of these stages, just in overview quite briefly, and then we'll look at this early stage once more. So in that research and development, 3 to 6 years, the drug development begins long before clinical testing. It starts with the identification of a target for a drug to act on, and then the identification of compounds that could potentially hit that target. So what will that drug affect? The target identification is key. Understandings of the causes of the disease or conditions can help researchers know what processes or pathways drugs to treat the condition need to be able to target. And in compound screening, this is where you have this large compound library of 10,000, maybe more compounds. And they'll be screened in laboratory tests for their ability to affect the identified target, typically a protein, say such as an enzyme. They're also screened to check that they don't interfere with other related targets, so perhaps similar enzymes. So which compounds to test? And that's in the lead identification stage. So through though screening is unlikely to uncover a perfect drug candidate, these promising compounds can be identified, and the structures of these molecules can be modified to try and improve their activity. So if you have now a optimized compound, then you will perhaps enter those into preclinical studies. So you would have screened initially, say, 10,000 compounds, but only around 250 will make it to preclinical testing. And this is where efficacy and potential risks are evaluated before human trials can begin. And this is often also seen as the lead optimization stage. So you'll be doing lots of studies. So you'll hear terms such as in vitro studies. And in vitro is Latin for in glass. So laboratory experiments on cells or molecules outside of their usual biological surroundings. And this gives some idea of the efficacy against the identified target. And toxicity can also be assessed. But typically this may be some assays where you are just testing a functional assay of an enzyme with a small molecule. You'll also be looking at in vivo studies. And in vivo is Latin for in the living. So drugs that look promising in vitro testing will progress to testing in sort of cell studies, or in particular then animal studies. These are the in vivo studies. These include tests on toxicology, efficacy, and how quickly the drugs are absorbed and subsequently eliminated. So then there will be also looking at toxicology, and to get approval for clinical trials on humans, you need at least 2 mammalian species. So drugs must undergo toxicity testing on at least 2 mammals, and 1 is a non-rodent, including at least 2 administration routes before they're allowed to proceed to clinical trials in humans. So this is a legal requirement. When you're in clinical trials, these typically take 4 to 7 years. So clinical trials involve human participants. These tests in volunteers provide information on safety and efficacy. And around 70% of drugs succeed in phase 1, 33% in phase 2, and then about a quarter, 25 to 30% in phase 3. So you can see things are dropping off through each time. So in phase 1 trials, this is usually, say, 20 to 80 people. The primary goal of a phase 1 trial is to determine the drug's side effects. Additionally, how quickly the drug is metabolized in a human and excreted from the body can be determined during these trials. And this is important to understand what dose you need. So in phase 2 trials, this is usually, say, 100 to 300 people. Phase 2 trials help to determine how effective the drug is in patients who have the condition it is trying to treat. Control trials compare the effects of the drug to that of a placebo. And in phase 3 trials, it's usually 1,000 or more people. And this gauges the efficacy, dosage, and safety in a larger population. It also compares the efficacy to existing treatments, as well as interactions with other drugs and effects of different dosages. So once passing all of these hurdles, then you'll need to apply for approval of your drug. And this can take 1 to 2 years typically. So if a drug is deemed effective in clinical trials, it is submitted to regulatory bodies to be approved. It's estimated that 1 in 5,000 drug candidates make it all the way to approval. So this is a very difficult process. So in the evaluation, they'll be looking at evidence from those clinical trials. The regulatory body reviews the evidence provided by, say, a pharmaceutical company for the drug's efficacy and safety. drugs offering important advances are given priority. So approximately 75% of submissions are approved. So most of things should be approved. And if the benefits of taking the drug are deemed to outweigh its risks, it's approved. The cost of developing a drug that gains marketing approval is estimated recently to be around $2.5 billion US dollars. So this is very, very expensive, but this is in some way taking into account all the failures that a pharmaceutical company has also been burdened with before it gets one approved drug. So once it's approved, there's also monitoring of the drug, and this is indefinite duration. So after a drug is approved and available for use by patients, it's still monitored for any side effects in the general population that may not have been occurred in the drug clinical trials. And there's been occurrences where drugs have to be withdrawn by effects that were not known at the time from clinical trials. So on this slide here, we're going to spend some time going through especially these earlier stages of the drug discovery process. So you can see at the top, there's two arrows. One is discovery and the other is development. So in fact, drug discovery involves both this drug discovery, perhaps earlier phase, but also the development phase, which typically is the sort of preclinical to clinical trials and approval. So let's think a little bit more about this whole process. So the drug discovery process is expensive and time-consuming activity, and it mostly fails. Retrospective analysis of the pharmaceutical industry estimates that each new drug to market takes an average of 14 years, and it costs billions of dollars. In addition, only 1 in 9 compounds that enters clinical trials makes it to the market. The attrition rate in discovery research is similarly high. Depending on the company, therapeutic area, and discovery strategy, at best, only 1 in 10 research projects that begin with a starting compound will generate an optimized candidate to enter clinical trials. For these reasons, most companies maintain a pipeline with a large number of projects in the early stages, taking a diminishing number forward at each stage. The discovery process gets more expensive as you proceed, hence careful management of the portfolio is essential. The key to make the right decision at the right time, knowing when to stop a project, is often more important than committing to continuing. Modern target-oriented drug discovery is usually organized into a series of stages, and the definitions of these stages can differ from company to company, and the details of the boundaries will vary from project to project. But of course, the first stage, as shown here, is this target discovery stage. So clearly, this is the starting point to identify that relevant target. And in a pre-genomic era, targets were discovered through cellular and protein biochemistry methods, where a detailed understanding of the origins of the disease led to the isolation and characterization of key molecules. But now there's more genomic research which has assisted in this process. So in biological research, it's been dramatically changing, and the successions of omics technologies become available. So this is typically what's used now to identify your target. So whatever the mechanism of identifying a target, there needs to be some level of validation before nominating it for drug discovery project. So the phrase target validation is much misused. A target cannot be said to be truly validated until a drug is actually uniquely affects that target and is on the market. But it is typically used in this stage to just be as sure as you can that this is an appropriate target to be used. So then we move to the stage of screening and hit identification and hit generation. So a hit is a compound that binds to the target and has the desired effect. The conventional method for identifying hits is by screening a compound collection, which could consist of natural products or substrate mimetics, legacy compounds in a compounds collection, compounds synthesized as potential hits against a particular class of compound, target, or commercially available compounds. The majority of large pharmaceutical companies have invested considerably in automating this initial phase of hit identification, both in the generation of suitable target libraries and in the initial assay. This high throughput screening, or HTS, approach places considerable constraints on the robustness of the assay and the available and properties of the available compound collection. In most cases, hit identification phase relies on configuring a particular assay to monitor binding or inhibition. Usually a large number of compounds are being screened, so the first experiment is to measure compounds that exhibit activity above a certain inhibition at a set concentration. This is usually followed by confirming the hits. It's where an in vitro assay is run at varying concentrations to determine the IC50, and this is the concentration the drug is required to achieve 50% reduction in activity of the target, usually in vitro, or the Ki or Kd of the compound, and the Ki or Kd is the inhibition constant for the reaction, usually binding to in a particular assay, say a protein. And so the compound quality is checked and the sample is checked. Maintaining quality in a compound collection is a major challenge because compounds decompose over time, particularly if held dilute in solution in air. So in addition, it's not unusual for 5% of compounds purchased from commercial suppliers to either be not what they claimed it to be or to contain major contaminants that give false positive or false negative results. So an HTS campaign can require significant resources of compound, target and manpower and last probably usually around a year. It depends how long it takes to configure a robust assay. So an output from a hit identification campaign is set where compounds of chemical structures have been checked and where they can reproducibly shown to have activity. Then we would move to a hit to lead stage. The hit to lead stage, or H2L phase, is where some of the crucial decisions are made in a project, establishing which chemical series has a potential to be optimized into a drug candidate. This is an important decision as lead optimization, the next phase, is where significant resources are required. So it gets very expensive. For these reasons, most companies set quite stringent criteria for entering a lead optimization for each target and reflecting a projected requirement of the properties of the final drug candidate, often called the target product profile. The detailed work of the hit to lead phase varies with the nature of the project, and in particular, the origin of the hit compounds. Whenever the compounds come from, it's usual to resynthesize the compounds for complete validation of the hit, to either repurchase or synthesize close analogues of the compounds. In general, it's during the hit to lead phase that dramatic changes in chemical template are made and the essential core of the lead series is established. Usual aims are to establish preliminary structure-activity relationships, or SAR, within one series or more series to exploit the indicative physicochemical and ADMET properties. So ADMET stands for absorption, distribution, metabolism, and excretion, and toxicity. So depending on the project and the company policy, entry into lead optimization can be gated by demonstrating some in vivo activity. That's activity in an animal. Setting the right barriers for entry to lead optimization is one of the most challenging aspects of medicinal chemistry. So usually around 6 months is taken in this phase, or up to a year, to establish the lead series and enter lead optimization. So lead optimization now is the most resource-intensive component in drug discovery, requiring considerable input from synthetic chemistry, modeling, disease biology, and assay design. Not only is it unusual for lead optimization team to consist of over 15 scientists, particularly if more than one lead series of compounds are being progressed. The main challenge is to develop one or more compounds with desired drug-like properties, as well as having sufficient affinity for the target. And usually a nanomolar inhibitor is required. The compound needs to have an appropriate selectivity profile and be able to get to the site of action, many of which targets mean cell permeability and have acceptable drug-like properties. In addition, it's important to continue to track the observed effect on the cellular and then later in vivo activity is in interaction with the identified protein target. Though in the end, the most important feature is that the compound works in the cell, and pharmacodynamic markers are important to check if the compound is affecting the biology through the predicted target, particularly when an understanding of the structure of that target is being used to guide optimization. So in the early stages where some modest changes in the central core of the compound, however, the lead optimization progresses, main challenges are on the periphery of the molecule. The main driver is the biology. It's remarkable how quite small changes in the chemistry can have a large effect on the biological activity, particularly in vivo. The lead optimization stage typically takes sort of 18 to 30 months, depending on the complexity of the target biology, resources deployed, and the chemistry of the lead series. The real challenge in lead optimization is balancing when certain properties need to be introduced and deciding when to abandon a particular project or lead series. And then you will enter these preclinical studies and animal models. And this phase is to prepare for testing the compounds in a man. This includes scale-up of synthesis, formulation, toxicology, and design of clinical trials. The difficulty and cost of synthesizing the compounds is considered throughout the discovery process, but becomes particularly important at this stage. A synthetic scheme that works in the laboratory to produce 100 milligrams of compound may need dramatic modification to produce the many kilograms of compound required for late-stage clinical trials. Overall, the difficulty of synthesis or purification of a compound will have a marked impact on the cost of goods, i.e. how much it will cost to produce the drug, and this can seriously impact the commercial viability of the project. Similarly, formulation, getting the drug into a form that can be administered both for the animal testing and for clinical trials, can have an impact on the project viability. This phase is to prepare the way for clinical trials where the drug candidate is given to humans. This is covered by a stringent regulatory regime, and many of the steps in the preclinical stage are covered by regulations and need to work to certain legal guidelines. Then we would enter the phase of the clinical trials, and this is the clinical full development of phase 1 to 3. So this is usually the most expensive and time-consuming of the overall process of discovering a new medicine. It's conventional to think of 3 separate phases. In phase 1, studies are primarily concerned with assessing the drug candidate's safety. A small number of healthy volunteers are given the compound to test what happens to the drug in the human body, how it's absorbed, metabolized, and excreted. A phase 1 study will investigate side effects that occur as dosage levels are increased. This initial phase of testing typically takes several months, but about 70% of the drug candidates pass this initial phase of testing. In phase 2, the drug candidate is tested for efficacy. So usually this is explored in a randomized trial where the compound or a placebo are given up to several hundred patients with the condition or disease to be treated. Depending on the condition, the trial can last from several months to a number of years. The output is an increased understanding of the safety of the compound and clear information about effectiveness. Only about 1/3 of the project successfully complete both phase 1 and 2 studies, but at the end of this process, the compound can truly be considered as a drug. In a phase 3 study, a drug is tested in several hundred to several thousand patients, and this provides a more thorough understanding of the drug's effectiveness, the benefits of the range of possible adverse reactions, and these trials typically last several years and can include comparison with existing treatments on the market to show increased benefit. These trials provide the necessary data on which to get approval by the regulatory authorities. As a drug comes towards and is launched on the market, continued trials and monitoring is required. Sometimes adverse reactions can only be picked up when a drug is given to a very large population. Problems can sometimes be dealt with by changing in prescribing practice or thorough defining patient populations. However, it is sometimes necessary to remove a drug from the market. So I hope this has given you a good summary of this journey of drug development, and I thank you for your attention. Thank you very much.
Great. Thank you very much, Professor Spring. That was a lightning presentation you gave. It takes me back during my MPhil studies at the University of Cambridge, where I studied therapeutic sciences, and then we got to learn and understand the intricacy of drug discovery and development in detail. So it's really good to think back and remember all that I studied. That's really great. And then thank you for your presentation. Please, does anyone have any questions or comments that you think you would want to ask Professor Spring? Anything you didn't understand, anything you need more explanations to? Both online and in person. If you are in person, you could come in front of the stage, take the mic, and then ask questions. If you are online too, you could drop your questions in the comment section, and I'll be happy to pin this and then let David respond accordingly. Is there any questions? Okay, if there are no questions, then I would say thank you very much, Professor Spring, for joining us. Thank you for your time, and then we wish you the very best for the day. Thank you very much. Yeah.
Thank you. Goodbye.
Thank you. Bye. So next on the agenda, I would hand over to my colleague, MC on ground, Amaka, who would introduce the next speaker to give us opening remarks. Thank you very much.
Okay, once again, good morning, everyone. Okay, I'm going to introduce Professor Ikechukwu V. Onishi, who graduated from the Faculty of Pharmaceutical Sciences, UNN, in 1995. He finished his master's in 1997 and PhD from the Department of Pharmaceutical Technology and Industrial Pharmacy in 2005. He was promoted to a professor in 2013 and has researched widely in drug formulation and delivery. He's an international expert in procurement and supply chain. He's the current Dean of Faculty of Pharmaceutical Sciences, UNN. He's ably represented by Dr. John D.K. Obona, the Associate Dean of Pharmaceutical Sciences. Let's give him a round of applause as he comes up to the stage.
Good morning, everybody. It's surprising that the hall is not filled up to the brim. We thank God that we are all here. I thank the chairman of AODI, Dr. Chooks, who has been my old-time friend since 2011, 2012, even before he left for his PhD in Cambridge. Chooks has been doing a lot of things for the university, and I'm surprised that this function wasn't really publicized. I heard of it yesterday, and I wanted to get in touch with him or Chiamaka, because we have some colleagues who are working together. Chooks, Chiamaka, Don Weibo, and Olika Ago, they're all in US. I'm happy that many of because I try sending this information to the class rep of the graduating pharmacy students and probably for those who are in 50th year, because Chooks has through his workshop on, when I say this is the second time he's doing it, because his plan is that it will be an annual workshop. And through this workshop, it will open more doors, linkages, and collaboration between our students, University of Nigeria, and in the United States of America, another part of the world. Because most of the people that are going to talk today are not just people from US. We have other people that he has collaborated with when he was in UK before going to US. And I pray that today, many of the students who are going to be here will really tap from the experience of a lot of the researchers we have here who are really experts in their different fields. Another thing I want to say is that let nobody go home without having any contact you have collected. I know that the slides are not too friendly because even where I was, I couldn't even see the slides clearly, not to talk of those at the back. So I just hope that there will be improvements in the slides so that at least one or two people can get contact of those who are there. And if by virtue of the exam that you now end up having a very good CGPA, either as an undergraduate or master student, you can always collaborate with some of all these people. It's not a rocket science. So don't just feel that because people that are talking are in US or in UK or from other parts of the world, that is something that's not doable. So by the time from this workshop, you are going to collect probably the email address of everybody, and they will share the contact of all the information that most of them are going to deliver today. From there, you can pick the contact or email address of all those people who must have spoken this morning. And again, don't feel demoralized, because sometimes you may send mails to whites. You'll be so surprised that the person won't even read your mail. You can send 100 mails, maybe only one person or two will read the mail. Because I believe, just like when we started this one, I was like trying to tell Chiamaka that these people believe in time. Because having worked over there for some time, I know that if they have an appointment with you by 10, you're supposed to be there 5 minutes before 10. Meaning by 10 you start. There's nothing like, let us pray, let us do this, or let us do this. They go straight to the point. So please, I'm hoping that everybody who is here must have dropped his contact so that by the time they finish, they will send all the information to everybody's email address. And from there, we hope that other things will open up for every other participant. Thank you.
Thank you very much, Dr. John. Thank you very much for your speech. As he's rightly said, most of us who you see here started like you. As I said, I was at UNN last year when we organized the first or maiden edition. As some of you might have remember, I said I started like some of you, but I was in Ghana. Okay, we all started like you, and then we had opportunities to travel abroad, to study, and then make things happen. So as you are there, don't get discouraged. There are lots of opportunities. You just have to work hard and ensure that you grab opportunities as they come. Take this opportunity to connect with people, those you haven't spoken to. I know that there is this saying in Ghana where people go through the university, but they don't let the university go through them. In this, we mean that people come to university, they work solo, they go and hide at a place, study solo. After exams, they go back to their hostels or hall of residence solo. They don't make friends, they don't involve themselves in any activities. And then at the end, they don't really know anyone, they don't have any contacts. If any opportunity comes, no one even knows them. You would have to go through the university, involve yourself, attend functions, network. Networking is very important so that you can even be introduced or be approached by someone who even once met you during an event. And that is how doors or opportunities open. I'm hoping that with this event running for the second year, you will be able to take opportunities to connect with each other and then grab opportunities as they come. So moving on, I would like to introduce the next speaker, who is basically one of you. He's going to take the stage for the hands-on workshop, unless we have the head of biochemistry around to also give his opening remarks. But if not, I would move ahead and then introduce the next speaker. Please do confirm on site. Okay, so silence mean consent. So if I'm not hearing anything, then it means he's not available. And since we are far behind time, I'll be moving on to introduce the next speaker. So it's a pleasure to introduce a talented researcher who will be leading the hands-on workshop today on molecular modeling and computer-aided design. Please join me in welcoming Samson Onoyima. Samson is a dedicated research assistant with the Entomology and Protein Chemistry Research Unit at the University of Nigeria in Nsukka. He is also an esteemed member of the Department of Biochemistry at the same institution. Samson obtained his BSc in biochemistry from the University of Nigeria in Nsukka in 2017. Since then, he has been contributing significantly to the fields of enzymology and protein chemistry, showcasing his expertise and passion for scientific research. His workshop today promises to be an informative and interactive session, providing valuable insights into the cutting-edge techniques of molecular modeling and computer-aided design. Participants are in for an engaging session that bridges the theory and practical application of scientific endeavors. Join me in giving a world welcome to Samson Onoyima. Samson, over to you when you are ready. Thank you. As Samson is getting ready to upload or show his slides, I would say so as speakers are speaking or presenting, please, if you have any questions, it would be good to scribble them down so that immediately speakers finish presenting, you can come forward and then ask your questions, and then this will greatly be appreciated. Don't wait till we start calling you individually. It will be good to have your questions down so that immediately the speaker finishes, you just walk up in front, and then you'll be able to ask all the questions you need. Francis, can you upload your slides, or do you want me to upload this for you? Okay, I think I'll do that. Okay, 1 minute.
Okay, while the upload is going on, I would wish to thank the organizers of this particular program. And for many of us that are here seated, is a very big thing to us. Number one thing is inviting people to come, and then another thing is them showing up. I'm so glad I'm seeing a lot of students as well here. I believe so much that many people are actually going to go home with something today, even as we proceed. Our deans, our lecturers that are here, we wish to appreciate you people for the time and the encouragement and all that. I'm quickly going to share a little of the slides. Just an introductory aspect of what we're going to do, after which we are going to switch into the full-time hands-on exercise. Okay. Fortunately, I was here last year during this same event, and I left with a whole lot of things in my mind, which I learned from the event. I hope that many of us here today as well will go with same testimony. Okay, quickly from slides. Okay, I'll be handling us on. Yeah, I'll be handling us on introduction to computer-aided drug designing and molecular modeling. Okay.
I think this is going to be a hands-on workshop. So please, if you have your computers available, it would be nice to bring them out so that I think Samson takes you through some hands-on demonstration in molecular modeling. Thank you very much.
Yeah, okay. Yeah, thank you so much. Yeah, please, for those that came in with their systems, the softwares we're going to be making use of in this particular workshop are all available and will be sent to you or installed to you afterwards or as the conference is going on. However, a link to all the softwares and their installation guide, I will make sure I provide that immediately after here for those that couldn't make come here with their own system. While following up the video later, you could be able to replicate similar thing you here. Okay, this particular introductory presentation is going to be very brief because the first speaker has actually made a lot of insight into the same topic, so we might not delay much here. Okay, my name is Onoima Samson Chineku, as earlier mentioned, from the Department of Biochemistry, UNN here. Okay, please, the next slide.
You can
Okay,
actually throw it
quickly
from your end as well.
Okay, quickly, we have few outlines we're going to look on to here before we commence the hands-on process. One is what is CAD, what is computer-aided drug design. Secondly, what is molecular talking? And then what are the steps involved in carrying out this particular study? Also, what is ADMET analysis? And lastly, what is molecular modeling, or what we call lead compound optimization? Okay, you can move on to the next slide. In the next slide, we have what is CAD, which I earlier stated. CAD, the full meaning is computer-aided drug design, and it's simply a computer methodology, okay, exploited in finding, developing, and analyzing medicines from be it bioactive compounds from plants or synthesized products, or what they call synthesized drug. Yeah, like some of us from the chemistry unit a few weeks ago were able to synthesize some novel compounds, okay, which they further investigated for their therapeutic properties. So these are one of the tools that can help you achieve that. Okay, CAD also is a methodology that helps to speed up the stage. Organize them instead of making fruitless efforts, testing a whole lot of compound and all that. Okay, quickly, we might try to compare, please, in the next slide. Yeah, quickly, we might try to compare the conventional method of drug discovery and then the CAD method or the CAD process. I won't spend more time here because it has already been talked about. Yeah, the conventional method tends to be longer, it tends to be time consuming and also cost expensive as well. Okay, from what you can see here, you have over 10 to 15 years just trying to arrive at a particular drug candidate and all that. However, the CAD process tends to hasten this particular process. Okay, you can move on to the next. There you will find two basic approaches to computer-aided drug designing. Trying to get a particular drug candidate which could further go into synthesis and clinical trials and possibly drug development. And it could go into different ways. The first is what we call the structural-based drug design. And then the second is the ligand-based structural drug design. Okay, in the structural base, which is what we're actually going to be focusing on today, it's trying to tell you or trying to look at having a 3D structure of a particular protein, out of which a particular ligand or a list of ligand could now be targeted at, okay, to ascertain a particular one that has more promising effect with respect to that particular protein. So here you have a protein, you have a target, and then you now go ahead to seek several ligands or compounds or synthesized products that could interact with this particular structure. Why the second one is ligand-based. Here you have your ligands, but you don't have your protein. Okay, you use the ligand you have to search for a particular protein that best fits that particular ligand, and as such could further be studied. Okay, look down, you will see a few steps in that might involve visual screening, selection of your compound, and then lead optimization, and finally your new medication is out. Okay, we could have a few advantages of a card, which we've mentioned a bit earlier. It's cost effective, it's time saving, it could give us an idea of the ligand receptor interaction. It could also minimize synthetic and biological testing efforts, which most times could be futile, okay? So this will help to minimize that. And also it minimizes chances of failures in the final phase of drug development. Okay, we can move on to the next.
Yes.
Okay.
Whilst we move on to the next 1 minute, I wanted to remind audience of this, especially those who just joined. So we have a science challenge which involve series of quizzes, series of questions. So these questions are taken from speakers' presentation as they speak. And then, so we would want you to please pay attention and then stay throughout the program, because the science challenge has an award. The first prize wins an amount of 20,000 naira. Second prize gets 10,000 naira. Third prize gets 5,000 naira. So if you would want to win these prizes, it would be nice to please pay attention and then stay throughout the auditorium, so that when we get to the science challenge, you could answer the quiz question and then get the money. Thank you. You could carry on now, Samson. Sorry for interrupting.
Okay, we continue. What is molecular docking? For the purpose of this card, we are simply going to be making use of these two, which we can actually have access to. Okay, molecular docking, like we have over there, is a key tool in structural biology and computer-aided drug designing. And then it helps to help us to actually predict a predominant binding mode, okay, or orientation of our ligand or our synthesized product or synthesized drug within our target protein, okay, or within our target, using the three-dimensional structure of that particular protein. I believe we'll get this more when we move on to the hands-on. The next slide, please. We might have types of molecular docking there. As you can see, we have protein ligand docking, which simply involves interacting your ligand with your protein. And then secondly, you can see that by your right in the first figure, you see a protein, you see a ligand, and then they coming together and giving you your product, a complex. And also we have protein docking, okay, where two proteins could come together. Yeah, two proteins can actually come together, when you are trying to investigate a complex and the effect of possibly a drug, why such protein exists as a complex. Yeah, we've carried out such researches before. Yeah, it is very, very possible. Okay, please, to the next slide. We also have, what are the tools, we can make use of in molecular docking. Yeah, there are varying tools you can actually lay your hands on or make use of while performing this task. And many are still evolving. The first we have here is orthodox vena, which most of us might be very used to or have heard about before. and which is actually what we're going to be making use of in this workshop. The next, we have Schrödinger Mistral, okay, from Schrödinger suite. Thirdly, we have molecular operating environment. This is quite a very expensive tool that most people might not have access to that. However, some of us do and can be given to us afterwards. Also, we have Pyrex, okay? Many may have heard about Pyrex as well. And lastly, you can actually use some online servers to perform your docking as well. Okay, one of them is PatchDock, as can be seen here. There are many of them you can actually make use of. Okay, let's move to the next slide. Okay, in the next slide, we look at the steps involved in molecular docking. Okay, we have the pre-docking activities, we have the docking activities, and then the post-docking activities, okay, which is about ADMET predictions and all that. In the pre-docking activities, you'll be talking about identifying your target and ligand, or synthesizing your product you want to investigate. Very, very important. The second one is the active site study of your target. Of course, you need to understudy the protein you want to investigate. Yeah, you don't just pick any target or pick any protein and or receptor and commence your investigation. You have to have some valid point why you're considering that. Also, you can go ahead to now source your target and ligands if you now have them handy. And then also you can go ahead to prepare your target and then your ligand. And lastly, on pre-docking activities, you prepare your config file, which we are all going to do stepwise here. In the docking activities, you simply commence the docking process, make few commands using this particular software, and you get your protein and your ligand joined together, where you can now visualize their interactions and all that. And then lastly, you could as well go ahead to look at some ADMET predictions or properties, or what we call drug likeness properties of your compound. Your compound might not just end up having a good binding pose or binding mode or good interaction. It should also have some good drug likeness properties. And then also pharmacokinetic properties as well. Does it obey the Lipinski rule of 5? Or does it disobey that? The course of the workshop, we're going to see to that. And then quickly, we might have few applications of docking. There are quite many, but I can only list a few here. Please, at the end of this, you are going to get the slides so you can, in detail, go through some of them just to gain a whole lot of things so we can quickly move on. Okay, please, the next slide. Yeah, you can go to the next slide. These are the applications we just talked about because of our time. Okay, we quickly look at molecular modeling or lead compound optimization. Yeah, when you commence docking proper, or when you start in silico studies proper, you'll discover that sometimes you might have a compound that looks promising. Okay, the compound might look promising. However, such compound might simply require some level of optimization or modeling, okay, to get a better version of the same compound. At the end, you might discover your compound is quite close to, the standard drugs that are already in the market. Remember, you are doing all this to, bring about a new therapeutic or a new drug. So at the end, you must try to see how your own particular compound could be optimized or could have some advantage over those that are already in existence. Okay, as you can see from the slide, this is an optimization study I did a few weeks ago. By the left, you will notice a particular compound. This is an anti-malaria drug I was trying to model here, or to optimize the compound here. I discovered that while this compound is located inside the active site or inside the binding pocket, there are some unexplored interactions, okay, of the same ligand with respect to the target protein. So what do? Being a computational biologist, I will look forward to how can I optimize this compound, to increase the interaction of this particular compound within the active site. Remember, this particular form of optimization is quite different from what many do, pharmacophore modeling and all that, where you intentionally add amino acids and all that, and all groups to your existing ligand and then trying to reduce that. That might just be a blind optimization. However, when you try to locate your ligand within the active site of your protein or your target, you could actually see the limited interaction or points of possible optimization you could explore to optimize your compound. That is exactly what I did here. And we can use tools like molecular operating environment to carry out this. So when you look at the first and the second, you can see that in the first there is a cyan patch, okay, a light green patch. I'm not sure if you can see that, but maybe when we have the slide, we'll see that. Then on the right side, you will see that there's now a pharmacophore, a circled something in purple color that have been located at that portion, that cyan portion. Cyan portion is an indication of a lone pair of electron, which is possible place of optimization. You need to make an interaction at that particular point if your compound needed to be optimized. So at the right side, you see me, exploring that particular point. And let's see what happens in the next slide. In the next slide, I've been able to, optimize my compound to make contact with that particular portion. You can see that in yellow. I try to use yellow to circle that. So he has been able to make contact with that. Then after this, I try to redock the same compound I've optimized into the same pocket and see if such interaction was actually visible. And then when I did that, in my next slide, you can move to the next slide. In my next slide, you see some dockings. Okay. Sorry, before this, maybe this particular slide is not there. Okay, but I try to add the binding scores because I feel I need to communicate to us the binding scores of the ligand after optimization and before optimization. Here I have a few of them. I have the model, which after binding, after the optimization and binding, was giving me a minus 12.1 kilocal per mole binding energy or binding affinity, while the original compound was giving me minus 10.9 binding affinity. I also investigated few malaria drugs, including artemisinin. Artemisinin is about 8.9 in binding affinity here, while lumenfantrine is about 9.4. Artemether is about 7.6. And then artemether lumenfantrine, I tried to also carry out a combinatorial therapy, a combinatorial docking here, where two drugs, sometimes we'll go to buy malaria drug, we'll find some in combination, something like artemether lumenfantrine. So it can exist that way. I try to also combine that way computationally and dock that. And then we'll find out, even with the combination, it's giving us about 11.9. However, my single optimized compound is giving me about 12.1. Possibly at the end of this, you get the updated slide and you see more of what I'm saying. Okay, we can now move to this particular slide. Yeah, you can now see the model and then the original compound within the pocket of the target or the protein. By the right, you will see one circled in green. If it's a bit clear to you, you'll discover there is actually an interaction that has been elicited there simply because of the optimization, which is actually lacking in the second one. Okay, we can move on to the next slide. Yeah, quickly, some people might be wondering, we'll be hearing about docking, we'll be hearing about molecular simulation. Are they actually the same? What is it all about? However, just to clear a little bit of that, molecular docking is a little bit different from molecular simulation. However, both are actually steps in drug development using CAD. Molecular docking is simply a basic study, which after that is supposed to proceed to simulation. Molecular docking can only occur using a rigid body, like we said, a crystallized structures or crystallized targets. However, in simulation, you go ahead to try to create an environment similar to that happening within the protein you want to target. For instance, if your protein is a hepatic protein, a liver protein, what do? While trying to simulate, you're trying to generate an environment similar to that which as when the protein is actually existing within the host. So you can actually see what happens while in motion, just like in the body, unlike the docking that purely occur at rigid body or crystallized structure. Okay, quickly, we can move to the next. I think I just highlighted as well the difference between the two. You can move on to the next. By way of concluding this little introductory aspect of the hands-on workshop, we discovered that molecular docking and simulation have greatly impacted several fields of scientific research. Okay, and as such should be optimally maximized, globally, to be able to have an improved research outcome. Okay, very, very important. Okay, we can move on to the next slide, which is, the next slide. Way of me saying thank you for listening to the little introduction. To the next slide, please. In the previous slide, you are going to find some of our contact, including our email address, and some other of them you are going to find after this workshop, where you can easily reach out to us if you are really, really interested in going further or taking some of these quite very serious. Okay, this is the hands-on exercise, okay? Please, those timing me should try as much as possible to be carrying me along so I don't
you have about 35 minutes, so still more time.
Okay, about 35 minutes. Okay, please, we're going to run along. Let's see, but I just try to lay some basis for us so we can actually further on. Okay, quickly, we are going to be looking at the next slide, please. Okay, we are simply going to be looking at the for the hands-on exercise. Let's see how this can be done practically. Okay, we're going to be looking at the anti-inflammatory effect of naproxen. Okay, naproxen is a standard drug, an NSAID drug that has been explored in treating inflammation. Most of us know what inflammation means. It's one of the system's mechanism of defense and healing as well. So in this particular case, we're going to see, sometimes we carry out some researches and we say our plant extract, has some therapeutic effect or have some therapeutic properties, such as anti-inflammatory properties, or you synthesize the novel drug and then you, say that such drug could actually have some effect. Remember, anti-inflammation is just one of them. You might have diabetes, anti-diabetic, you might have antioxidant, anti-osteogenic, and many of them. But for the sake of this, we are simply going to concentrate on the anti-inflammatory effect. And then in our next slide, which I was thinking I'm actually going to go live here so that I can actually carry us along very well. And please, the technical team, I think at this point, at this point, I really need help here.
Yeah, is it a slide you are going to use?
So the hands-on is not going to be slide. It's going to be real life experience. So I will need to actually log in here live so they can actually see the demonstrations while that goes on. So I don't know, please, those in charge, you can come closer and make sure that is rectified here. Okay, meanwhile, we are going to be investigating this effect using a particular target, okay, which most of us know as cyclooxygenase 2. Okay, I've mentioned earlier, while trying to carry out a particular study, you don't just go and pick a particular target or go and pick a particular protein. In doing this, we target or we look forward to the key enzymes or the key receptor or the key targets in a particular metabolic pathway or in a particular reaction, such that when we are able to interrupt such particular targets, we could end up achieving our goal, either via inhibition or via activation. So in this particular case, we considered COX-2 enzyme, which is cyclooxygenase 2. Cyclooxygenase 2 is a key enzyme in prostaglandin production. Yeah. Okay, please, a little while the technical team tries to get us on.
I'm thinking with a particular platform we are using, the StreamYard, it doesn't really let you share your screen directly unless you upload it on the platform.
Okay, I think we are trying to rectify that now. Click on it. Okay, I'm trying to see if I'm on now. Okay, those are the other end. I think I'm trying to share the screen here, but you might need to enable me. Okay, I think I'm on now. Okay. Finally, we are here. Okay. Okay, so yeah, that's my screen. When we'll get to that, this was what I was mentioning earlier about the scores. But let's continue with the hands-on exercise. Yeah, like I mentioned, this is COX-2, which we are going to be targeting or be considering in this particular hands-on. I mentioned earlier, COX-2 is one of the key enzyme, or cyclooxygenase. It's one of the key enzyme in inflammatory responses. Okay, it initiates the conversion of arachidonic acid to prostaglandin. Okay, and also note that this particular protein is bifunctional in nature. And one of the reasons why I considered that, so I can as well communicate this little trick to us. Some bifunctional, proteins or targets might have two. When you say bifunctional, I simply mean it has two binding sites, okay, or two binding pockets, okay, which are actively involved in a particular, particular pathway or metabolic process. In this case, we have about two major domains here, the membrane binding domain and then the catalytic membrane or the peroxidase binding domain. In the membrane binding domain, that is where the first reaction takes place, which is the key step of this particular reaction, where it happens. And I said this involves the conversion of arachidonic acid to prostaglandin G2, which is just an oxidation process. However, the structure of this particular binding domain has a way it connects to the next pocket. If you look at the structure, you discover that you have two red residues here. The first one, you can see me touch, is the membrane binding domain, where therapeutic investigation is always targeted at. While the second one, if you are conversant with some of this structure, you discover that this is a heme group, okay, which is usually present in any peroxidase pocket or kind of reaction. It's very involved in such interaction. So here, if you blindly carry out docking using this particular tool, where the grid setup is going to generate automatically, is going to be at the peroxidase binding unit or binding pocket, which is actually the wrong pocket. That's not the rate limiting step or the key step there. The arachidonic acid need to be converted to prostaglandins, which you need to stop if you're actually targeting anti-inflammation. Once that is produced, it could go ahead to be reduced at the peroxidase binding unit. As a result, you don't target that point, rather you target the first point. So I just showed us this because if you pick this blindly to dock, your default setup will go and locate the peroxidase binding pocket, and you go ahead and dock the peroxidase binding pocket, thinking you're actually doing the right thing. However, those that knows this properly will tell you that is wrong. Quickly, let's move on to the live demonstration of this. Quickly, we are going to commence the different steps I mentioned during the introduction. One is sourcing your proteins or your targets, and then secondly is sourcing your ligands, or if you had a synthesized product, is at this point that you bring up the product for your interaction. So to sort our protein or our target, we do that using one of the major servers, which is a protein data bank server. Okay, I can quickly locate that by simply typing PDB. Okay, once I type PDB and then hit my enter button, load, and of course we'll go to the home page, which is the first option here. I'll click on the home page and then that will load. Okay, when it loads, you discover an interface, which is the home page. One of what you see here, if you can see my mouse, is search button, where you can search for the name of your target, or if you have the ID of your target, you can go ahead to search that. Every protein crystallized and then uploaded is assigned a particular ID number, just like compounds as well. Okay, so if you want to locate that particular protein, you either search that through the number or the name of the protein. However, when you search by name, you are certainly going to find lots of the same protein crystallized by different researchers and uploaded there as well. However, when you search with a code, you simply locate your particular protein of interest. I've also mentioned you have to carry out some basic research before going to look for that. You don't come here blindly. You must have got what you're looking at. So here, we are simply going to be looking at the COX protein, which I mentioned. And I already have my code as 3Q7F. Okay, that is the code of the COX I'm going to be using. If I hit on that, it's going to load. Okay, let me come. Now I have 3Q7D. Sorry. Let's use 3Q7D. Okay, this is that. Okay, this is the structure of R-naproxen bound to mCOX. Okay, mCOX-2 enzyme or target, as you can see that in the board or in the slide. Okay, because of our time, I might not have all the time to explain a whole lot of things here. But if you go down this page, you are going to find a lot of information regarding this particular target, including the structures, the co-crystallized groups or ligands bound to them. You can see that as I move down, and many other information here. So the next, we go ahead to download this particular target so we can commence the rest of the process. You go to download file. By the right side, you can see my mouse. You go to download file, and then we don't download as FASTA sequence. FASTA sequence will give you the amino acid sequence of the protein. Rather, we'll go to the PDB format, which is the protein data bank format of this protein. I'll hit on that to download, and then it has downloaded from the top. You see that downloaded. I can now hit on that to open the protein. I already have the software installed in my system, so I can easily open that. You can see the color. Let me adjust the color to a white background so you can be able to see that a bit more clearly. Okay, so this is our protein. You can see that. This is the protein. Okay, we have our protein. The next thing after getting your protein is to look at the protein. What is it looking like? As you can see, I have a dimer of this particular protein. There are actually 2 subunits here. And how do I note that? Assuming I haven't come across this before, if I go to the right side, the left side of this interface, there's an arrow pointing towards there. I just clicked. Okay, the arrow will expose to you the content of what you have in this particular page. Okay, you can now see I have alphabet A, B, A, B, A, B. This A, B is trying to tell you that you have two subunits there. A corresponds to the unit A, while the B corresponds to the word unit B. However, you have three different A's there and three different B's there. The first A will always correspond to the protein or the target. If I click on it, you will see the protein highlighted in the structure. Okay. While if I want to get the second B or the first B, I'll just click on it as well. You find out it's the second protein. Meanwhile, the second A will stand for the ligands. Okay, you might not be able to. Okay, let me highlight this so that you see what the ligand stand for. Okay, you can see this now. The second A stands for the ligand. These are all the ligand group attached to the corresponding to group B and all that. So in this particular case, I don't need all these ligands here. So what do I know? I need to clean up this particular protein. I'll remove certain things here, including the co-crystallized water of crystallization. Okay, I think I mentioned earlier, in trying to elucidate 3D structure of the protein, there are a few crystallography. There is NMR, there is electron microscopy. However, X-ray crystallography has tried to stand out because of the resolution of the 3D structures always obtained after such taking note of while trying to assess or make your choice in picking a particular target from X-ray crystallized products. Okay, so what do I do here? I need to clean up some of these in my protein. I can't dock with the two of them. It will make no meaning. I'm only interested in one, but the two are actually exactly the same thing. So what do? I'll take off one of the units and concentrate on one of the units. So to take off that, I'll simply click on the protein. If I click on the numbers here, as you can see, it will simply hit on delete, and this has gone. And then I'll move to the next B, which I mentioned stands for the ligands. I'll simply click on that as well and hit my delete button, and that will go. Thirdly, I will hit on the P. The third P stood for the water. The first is your protein, the second is your ligands, and the third is your water. Remember, you could have more than even two subunits while trying to carry your study. So I can simply hit on delete. So I now have only the proteins here, only the protein of chain A, okay, or the unit A, which is what I'm going to work with. However, I still have water of crystallization here, which I don't need. So I'll go ahead and take off the water of crystallization. Okay, you can see there is no water here again. However, my ligand are too many here. Okay, I have lots of ligand here. You can see many of them. Sometimes while trying to elucidate the structure of your protein, some of these could simply be added for stability purposes. Okay, you try to stabilize the protein during the elucidation process. However, if you want to make use of such thing, you are still very free to take them off and continue. Okay. I need to take off all this. Remember, my is in the which is therapeutic target was not the if you go them, okay, it's my that's the one on the center is the biggest there. That's the heme group. If you have come across that before, that is the binding unit for the peroxidase pocket. While the next one close to it, yeah, you can see that is the membrane binding domain. This is where we're interested in. What you're actually seeing here is the bound neproxen we saw in the name. Remember, you have neproxen bound to the cog. So this is actually the neproxen bind. And then some of these you can find around are other groups, other ligands that probably during the elucidation process were there for some other purposes such as stability and all that. We need to take them all off. However, also for the sake of this, I wouldn't want to make use of this particular ligand or this particular neproxen that is co-crystallized with it. I would want to assess it in the I would like to access it individually in its own server, so that when you're now working with your own drug candidate, or you're working with your own extracts, you should be able to know where to find your ligand, okay? Not just the co-crystallized ones. So what do? I can simply knock off all the ligands that I have here. Remember as well, you can take them one after the other and remove them, but because of our time, I'll knock off everything that is here. So I'll bring back my protein. So this is my protein, or my target, which we now call the clean protein now. So I can go ahead and save this. I'll go to my file, I'll go to my save as. Okay, and then the window will show up. You will see the name which you are going to save. Then I can save this as my clean protein now, just for clarity purposes. Okay, then below it, you are going to see the file format of what you want to save. You have the Discovery Studio file. However, I'm working with a structure from the protein data bank. So whatsoever structure I'm going to be interacting with here is going to be in PDB file. So I'll simply change this to what? The PDB protein data bank file. And then it's saving to download. Okay, I will save to download. Meanwhile, immediately after saving this, I need to go to my download and make sure I rename or find a folder where I can quickly push in all our working materials here, so they don't go ahead being scattered and all that. So I will simply title this AODI Workshop. Okay. And then quickly, I will have to take my protein. Okay, let me copy this to my downloads. Okay. So this is the folder. This is my clean protein, as you can see from the top. So what do? I will cut that or copy that to the folder and make sure it's saved there. Okay, so this is our protein. We've got our protein. Let's quickly pick our target while we commence the docking process immediately. To pick your target, you make use of another compound saver. Remember, the first was for your target. The second, for your ligand, we get that one of the server we use is PubChem. Okay, just like PubMed is for your literature or literature review, your PubChem will take care of your words, your compounds, and their structures. So I'll simply type PubChem. Okay. If I type PubChem and hit on enter, it will open this way. And then the first option, I'm going to click on that. And then this is the home page where it's going to load. Then quickly, let me copy the name of my ligand. Okay, I'm going to copy the name of the ligand, which is N-proxen, naproxen. I'm going to copy that, and I will search. At the middle there, you see your search option. I'll simply place my search there and hit on my enter button. And then this will load this way. As you can see, this is the ligand, which is the naproxen drug. You can go ahead and hit on the name of the drug. It will open up to this. And then you can now go ahead to download this. All you have here is information concerning your ligand, okay, which you can actually explore later while doing yours. Let's quickly download this because of our time. If you scroll down, you'll find the 3D conformer. Remember, our protein was elucidated in 3D format. So your ligand as well should be in 3D format. Meanwhile, when you synthesize a drug, for some of us in the chemist unit, your drug, the one you synthesize or the one they identify for you, will always come with a structure. The normal structure draws for you is the 2D structure. Okay, you can use that directly and interact with a 3D protein. Rather, you will have to convert the 2D from 2D to what? To your 3D format. There are many tools you can use to do that. One of them is your Chem 3D, which is another package inside the Chem Office or Chem Professional suite. Once you copy the same compound, paste in your 3D, and save it back as what? 3D. You have just converted that, and then you can go ahead and work with it. So we can go ahead to make our download. You simply go to the download coordinate. You see that by the right side of the structure under the 3D conformer. Then under the saving, you won't see a PDB file here to save. Rather, you see the compound in SDF format, JSON, XML, and RES. we will simply download as SDF file. Okay, click on save and it has loaded. However, this file is in 3D but is in SDF file. Remember, we are working with protein data bank file, which is PDB file. So as a result, I will have to reconvert from SDF back to what the PDB file. Also, you have several tools you can use for this. Some tools, Pymos, OpenBabel, and all that. But for the time we have here, we'll quickly do this online and then we move on. To this, there is a server I usually make use of online to simply do that. It's more interface friendly and can easily help you convert that. If you type convert SDF to PDB, okay, and then you hit on your enter, you're going to see some options, online servers or options that can help you make your conversions, okay, and all that. However, I usually make use of the online Smile, okay. If you as well add convert SDF to PDB, online Smile, it will simply take you directly to the online Smile. So I'll simply click on the online Smile, and then that will load quickly. This is the interface. By the left end, you're going to see choose file. Okay, if you can see my mouse, yeah, that's the choose file. That's where you choose the ligand you want to reconvert from SDF to what PDB. So I'll simply click on the choose file. It's going to take me to my downloads, which is where my ligand is. Remember, this is the ligand. It was saved as Conforma 3D, okay, because it's an SDF file. So I'll simply click on that, and then it has loaded here. Okay, you can see that loaded here. The next thing, by the yellow window, you see display screen and all that. I'm going to choose my PDB file there. There are SDF file, we are converted to PDB, so I'll simply pick my PDB. And then by the other right end, you are going to see convert also to 2D or 3D. It's already in 3D, but here it was selected 2D for me, so I need to highlight the 3D as well to maintain the structure. If this is done, I can now go and hit my translate option. So I'll hit on translate, and then it's doing the conversion immediately. Okay. Okay, we are having some internet bridges here. Okay. Okay, it's fine. For the sake of this, once you click this option, it's going to, maybe the network itself is not quite but for the sake of our time, we'll have to fetch an already downloaded one just to help us complete this before our time is up. So I will quickly go to, I think I have that downloaded somewhere. Okay, so this is it. So I'll go to my downloads where I have the workshop file, okay, and then I'll paste this. That's all. Once the page loads, you will see click here. Once you click there, you get the compound downloaded. And when you download it, you rename it to your what? To your ligand. As such, it's now in form of your what? Your PDB file. So by the window here, you will see our protein and our ligand. So we've cleaned up our protein and our ligand is already clean. We don't need to clean anything with ligand. So quickly we go on with the little preparation and then the docking process very, very quickly. So to do that, we are going to be making use of the AutoDock to quickly prepare our compounds and protein, and then Vina for the docking process, and that should be all. Okay, tool is loading. Remember, the file or the link to this will certainly be given to us afterwards. You can easily access that later and commence yours as well. Okay, so our tool has loaded. So quickly, this is the tool we're going to be working with for the little preparation we're going to be doing before the docking. So quickly, to load our targets, the first thing we prepare here is our protein. So I will load that in, okay? And then you will see option like read molecule, you see option like read section. In this case, we are going to read molecule, not section. We are looking for a molecule. The protein is called a molecule. So I'll click on that, and it's going to take me to my my protein, or to the ligand, the protein and ligand folders, okay, where I can now navigate back to where I am. So I can now locate my downloads. I remember we have AODI workshop here. So this is the protein, titled clean protein. So I'll just click on that, and then it will get loaded here. As you can see, the protein. This is the protein loaded. You might not see that very visibly. So this is what we'll have. The first thing we do here is to color the protein, okay, so as to be able to see the different atoms or units in the protein. And I'm coloring this by atom type. If you look at the pop down from where I clicked, in the last angle here on the downward, you see a pop down menu, which is what where you can type or color your protein. So click on by atom type, and then I will say close. Well, noticeable enough, you discover, maybe because of the slide, you discover some color changes occurred in the protein structure, such that the blue coloration represents your nitrogen group, the ash colors are for your carbon groups, the whitish color, which are not much here until we add them, are for your hydrogen, yellow colors are for sulfurs and all that. So quickly, we have got our protein here. The next thing is to now edit our protein. Remember, the process of crystallization of this protein is possible. Some groups of this particular protein, or target has been removed just to enable the elucidation process to commence. So one of that is some charges or some hydrogen groups or hydrogen new atoms, which are quite very important in the interaction or docking process. So what do? I'll go to the edit button, still in the upper part of the options here. And then the next thing I'll do is to add my charges. You can compute your charges, or you can add your charges through add Coleman charges, or you can use the compute Gantt integer. Most of these does similar or do similar word function. However, I usually make use of compute Gantt integer. So I'll simply click on that. It will tell me that a total charge of about negative 1.9958 has been added. I will say fine. I might not see that visibly. Okay, the next thing I do is to add the hydrogen group that may have been lost, especially the polar ones that may have been lost at the course of the crystallization or the structural elucidation process. So I'll do that by going to edit, go to hydrogen, and then you will see under hydrogen, once you click on that, it will pop up. Then you see your add there where we can add hydrogen. In the add section, you can go ahead and click on it. And then when you click on the add section, a pop-up menu will appear trying to tell you, are you trying to add all the hydrogen or just the polar ones? Yeah, for the sake of this, we are going to make use of just the polar ones. Adding all hydrogen will make the whole structure quite populated, and clashes of interaction at the process of your docking study. So we select the polar ones only. And then I can click. If I click OK, if this is OK, well, when I click OK, the hydrogen will get added. With time, maybe we'll get to join back. Okay, I've been able to add the hydrogen as well to the group. The next thing I do is to generate what we call the grid setup or coordinate for my interaction. This is where the protein or the ligand is supposed to locate in the protein and interact, which is what we call the binding pocket as well. If you do this blindly, you might end up having your ligand binding to all sort of pockets anywhere it sees a hole in your target. I've come across a document where the researcher had pocket 1, pocket 2, pocket 3, pocket 4, for your ligand binding, which is actually wrong. So the person virtually did a blind docking, and then your ligand were just binding anywhere, which is quite wrong. So in this next option, we are going to locate our pocket where the ligand is going to be directed to bind. However, okay, we are back. We are back. So I was trying to show us in the structure. You can now notice some white portions of the structure. Those are the hydrogen atoms we just added. So quickly, just for us to be following, let's go to the grid now, which is where we can commence the next setting up of our protein, our target. So if you go to grid, which is a bit below some of the options there, you can see that where I am clicking. So if you go to grid, you can go ahead to, first of all, save this particular protein you've been able to generate. Now, remember, you've been able to edit it, add hydrogen, add charges. So you now have a kind of a complete protein now. So the next thing we do before generating further coordinates there is to save out this particular protein and know, okay, this our protein is set. Then we can now add the remaining coordinates where we are going to be targeting. To do this, you go to your grid, go to your molecules, and then you choose your molecule. Okay, if you click on that, by your left, you see an option coming up. You simply click on the option which has your protein name. By that, select molecule will get highlighted. Then you click on your select molecule. This is supposed to tell you a pop-up, which is initialization, and also that your target contains no non-bonded atom. It means that all your atoms were properly bonded, which is very fine. So you can go ahead and click OK. This will take you to your folders where you can now save that. We have ours in the downloads. Quickly go to my downloads. I'll locate the AODI workshop. Yeah, it's telling me no item matches my search, which is because the format of this tool I'm using to prepare this is in PDBQT. Orthodox VNA doesn't understand other file except in PDBQT. So if you save yours as a different extension, certainly it won't dock. You keep having error messages and you might, get confused how that came about. So we simply click on what save and we discover that this file has been saved. So the next thing is to generate the coordinates for this particular protein. So what do? I will simply go to the same grid. I'll go to, now I'm done with macromolecule. I'll go to set map type. Okay, we're trying to locate a portion now. So first of all, set our map, and it's going to be done directly. You just click on that, and by the top, you will see some, little pop-up telling you to accept the map type they have by default generated for you. Say yes, you accept this. And then the next thing, you still go back to the same grid. By this time, you are going to grid box. Okay, if I click on this grid box, if you can see that portion, something is going to appear there. Once I click on grid box, something appeared in the pocket. And if you noticed, or when you'll be doing yours, you notice that this portion where this box appeared seems to be the center, not just seems, actually is the center of that particular target, which is where the peroxidase binding unit or binding pocket is, not the naproxen binding pocket. So if you simply copy out these X, Y centers here and Z centers here for your docking, you are simply interacting directly with the wrong pocket, not on the main pocket. So what do? We'll have to change this to our own. Okay. Okay. I am simply going to round off from here. A lot of challenges came up while trying to, get this done earlier. So, however, wherever we stop here, at the end of this, I'm going to share to us some link that from A to Z of this has been, done. If you are really following and you want to continue from here, time will not permit us to take this to the end, as I have been alerted now. Is going to be made available, and we'll simply have to round it off here, and then but quickly, let me summarize here. I was trying to tell us quickly here that this particular portion is for the peroxidase unit. So the next thing you do is to locate that of the naproxen unit where you're supposed to bind, which is the next step you are going to take. So when you locate that unit, you discover that this particular box here will change. When you start moving this, you discover that that particular box here is actually moving along. The same with this. So this will help you to locate the pocket where you are supposed to bind this. And how do you do this? You simply do that by doing what we call site-specific molecular docking. Site-specific molecular docking will enable you to target a particular residue in the pocket where you want your protein. And from there, you can be able to target that particular protein. We have arginine 120 as our target amino acid, because this amino acid is very much, that's the amino acid in the triad of this neprotein binding that is responsible for binding drug. So what we do and make sure we block the amino acid. When you block that, further entering or interaction can't occur. So Gauss view will help you to open up the protein so you can locate the atom amino acid 120 and be able to exchange the coordinates for what you have, generate the same box and go and continue your docking process. We might just have to end that here, and then maybe through the links you can go ahead and complete your study. Thank you so much for your time. Okay, I don't know if I'm to take questions now or it's going to be afterwards, the moderator. Is the moderator still there?
Okay, thank you so much, Samson. You can go ahead and take questions.
Okay, please, very quickly, if you have questions with respect to I see someone raised, maybe those down there can help us. Let's get maybe 1 or 2 questions because of our time, and then we can wrap it up.
Okay, 1, 2.
Someone over there. Is here an additional mic? I think someone over there is not talking. Okay, you can use this.
Please, if I give you a number, just come forward, please.
No, they can come forward. We see the
camera. Come. Someone there too. Okay.
Okay, sir. Thank you for the lecture, the workshop. It was very insightful. My questions are these. The first one is, is there any tool or database where you can get information about ongoing or completed chemical trials against a particular target protein? So that is the first question. And then the second one is, when you were doing the workshop, you removed the water of crystallization. So I wanted to ask, is there a set rule on deciding the addition or removal of water molecules during ligand preparation? So these are my questions.
Okay, very quickly, the second question, the ligand comes out as clean as it is. So there is no water of crystallization with respect to your ligand. Okay, is your protein, while getting that elucidated structurally, there's always water of crystallization because of the crystallographic method you are making use of. And then firstly, you said if there is a site or a server where informations about ongoing field in silico studies are being reported, right? Okay. Basically, PubMed could be of great help here. When I was working on this anti-malaria modeling or lead optimization, that was where I came across some research by some kind of groups in India. They have a compound they're optimizing for malaria treatment. However, such companies still on that trial, so it hasn't really been So I just decided to exploit that particular privilege and look at that. So PubMed could be of help. And then even Google, many other, but most times your PubMed has more of likely information to provide to you. Okay.
Okay, good morning, everyone. Thank you so much, sir, for the lecture. So my question is more like to get more clarifications. So the sole purpose of molecular docking, if I got that correctly, it's to get a target drug, per se. So firstly, so how do you go about knowing that you need to actually start exploring on this particular compound to the point of now knowing that it's a drug candidate? Yes.
Okay, quickly to attend to that, like I mentioned, molecular docking is simply a basic step, is one of the number one step, okay, in trying to investigate the drug candidacy of a particular active ingredients. We usually call that active ingredients if it's coming from plant extract or a synthesized compound or product from the chemist unit. So what do you do after molecular docking? There are some stages where you're supposed to incorporate some standard drugs, okay? While investigating your compound or your synthesized product, there is need that you investigate that alongside the co-crystallized ligand, which is the native ligand of that particular target, and some already existing standard drugs in the market. All these drugs you take today, if you go to the PubChem server, you can download them, as a ligand and work with them. So for you to know that your compound or your active ingredient is doing well, you need to compare what it has based on one, the binding score or the binding affinity, and the mode, the binding mode or binding orientation within the pocket of that particular target when compared to standard drugs or the original substrate or ligand of the compound. That will now tell you if you could go ahead to optimize that further.
Okay, good day, everybody. So please, my question is very simple. Something caught my attention when you were talking about the components of protein before the docking. So I want to know, because you said something of coloration before docking. So I want to know, is there a way to determine a particular color for each component of that protein before docking, like the hydrogen, the carbon, and the rest components? So if there's a particular way to note that a hydrogen is supposed to have a white color, you understand my question, sir? Okay. Then another one is this. Please, is there a way, because I don't think molecular docking is being adopted in our degree level courses. So is there a way we can actually help or advocate for this particular course to be taught in the university as a topic or as a course? It will go a long way in helping some of us who are interested in things like this to start practicing before getting to where we want to go to. Thank you so much, sir.
Okay, quickly to your first question, how do you know the color of your protein? If I tell you, okay, this is the color, I only knew that because I have known what protein is before and I've studied that before. So when you research about your protein or you come to hear about proteins exist in their 3D forms. In 3D forms, they could exist as ribbon view, okay, as cartoon view, as surface views, and sphere and all that. In your ribbon view or cartoon view, you discover that there is something we call the alpha helix, and there's something we call the beta pleated sheet, okay, where the alpha helix by default is always colored red, and then the beta pleated sheet on your blue, and then some other loops on the protein or target having different color. These are by default, for a protein existing in ribbon, this is their default color. The same way when you load your protein in.
Okay. Yeah, when you now load the protein in tools like Orthodox tool, it will expose the different components of the target, not just the alpha helix, the beta plated sheet, and other loops. It will expose the different atoms by default, except you go ahead to alter the configuration, the color setup of the tool. It has by default set up the ash colors to represent the carbon copies or carbon groups, the whitish groups for your hydrogen, and then the oxygen in red colors, your nitrogen in blue, and then your sulfurs in yellow. By default, just like your ribbon 3D view occurs in default. That's how that does. So you can simply write that down, or you could go ahead, However, you could alter this configuration. Like I mentioned, this is just their default setup. Then lastly, your question, if you permit me, I will direct that to our dean and then some of our lecturers that are here to help us attend to that particular question. If it's going to be inculcated in our undergraduate curriculum, is going to be their task to do. However, many of them are already exploring this particular study. Yeah, many of them from biochemistry, chemistry group units have already started during their undergraduate defense, ensuring that their students go further to carry out in silico study, molecular docking study of their compound. I work with many of them this past few weeks that investigated such papers. So is almost getting into the system. However, when and how will be better addressed by our lecturers, especially our dean that is here, and the rest of them. I hope that's better for you. It's fine.
Okay, so there's also a question online for you, Samson. Enoch is asking, please, I wish to ask why proteins are often used as drug targets.
Okay, why proteins are used as targets? Is that your question?
Yes, that's a question from Enoch Arnold.
Okay. Well, sometimes few persons or most people interchange protein for target. You see people talking about target protein. Okay, in computational biology, we simply title that or term that your target. Your target could be your protein. In fact, protein is the umbrella name because even your receptor, okay, is still a protein. Okay, so when you call it a target, it's better addressed as a target. Target could be your protein. It could even be your RNA. Yeah, sometimes you can target a particular RNA for docking study. So your target is what we call protein, receptors, RNAs, and any kind of structure or macromolecule you intend to target for a particular investigation. So your protein is actually a target, okay? Okay, the moderator, do we get a response?
I heard there's one more question from someone.
Okay.
Good afternoon, our facilitator, and good afternoon, everyone. My questions, starting from the one I think is the easiest. Carbohydrates, can molecular docking be carried out for carbohydrate against a protein? That's first. Then secondly, let's say you are carrying out an experiment and you come up with a novel structure or a novel substance, for instance, and you want to carry out an in silico study for that novel substance. Let's say the structure is not on all these websites where you can get structures. So how can you derive the structure for that particular novel substance that you want to dock with? Then lastly, molecular docking. Let's say you are trying to compare your substance or your protein against known drugs. So because I want to know at the end, how will you infer that this thing you have now is actually can actually be seen as a drug, or everything you have been doing is actually successful, because you were trying to go to that before you were now saying there was no time. So I want to know how you infer at the end of the day everything you have done if it's successful. Thank you.
Okay, quickly, the first question is if carbohydrates could be targeted against a protein. Yes, it's very, very possible, be it carbohydrate, be it lipids. Yeah, something like arachidonic acid is actually a lipid, okay, and is even the main substrate of this particular target we made use of, if we can recall from the beginning. Cyclohexene converts the arachidonic acid to prostaglandin G2. So the substrate here is prostaglandin, is the arachidonic acid, not even what a ligand. So you could use carbohydrate, you could use toxicant, you could use lipids to target a particular protein. It's very, very possible. Then the second one is, yeah, if you don't have the structure of your compound on the server, okay, what are you going to do about it? Yeah, quickly, you saw me trying to open one of two here. This is ChemDraw Professional, and most of us might come across that before. I have come across that. We as well have that. We can share that with you people afterwards. You can actually draw the novel compound here. Okay, you simply copy the structure from the characterized compound. If you synthesize a new product, you go for NMR characterization. That characterization will elucidate the structure of what you just synthesized in 2D form. So you can actually carry the structure of that particular compound and paste on your ChemDraw Professional, or you could go to structure under ChemDraw. If you move down, you are going to see convert name to structure. It might not be that visible, but you're going to see convert name to structure. If you have the name of the compound as well, you can quickly, input the name here. If I hit on this, it will give me option where I can insert the name of the compound and then click. It will generate the structure of that particular compound. And once that is done, you quickly go to your Chem 3D, okay, which is here, and copy the same compound and paste here to convert from 2D to 3D. And then that attends to that. The 3D will also help you. If you go to your save as, you have option of saving as PDB, even from these 3D and all that. And that settles for a novel compound. Then lastly, I think you were trying to ask, how do you know your compound has met some criteria of being a good drug candidate? Was that the last question? Okay. Yeah, sorry, the time couldn't permit us to go a bit in depth into this. Yeah, before you can say, yes, your compound has met some criteria, of course, you have to, first of all, carry out the docking process. Remember, I mentioned while you are doing the docking, you are as well comparing what you're doing with some standard drugs and the original substrate or ligand of that particular target. How your ligand is actually performing. If at the end, through the binding energy or binding score is one of the easiest to compare how your compound is doing. If at the end, your compound could, number one, show good binding score or binding affinity when compared to standard drugs, or when you now try to visualize the interaction, your compound couldn't lay much reasonable interaction, then there is no need going further to optimize the compound. However, if you still have hope or trust in such compound, you could go as well to do what? Possibly optimize it and see what happens afterwards. However, after optimization, you still need to pass it through these steps to enable you see how that perform. Remember, I also mentioned that docking is just a preliminary step. After that, it's supposed to go ahead for simulation. It's the product of the simulation that should give you a more insight into the behavior of this your ligand or drug candidate, even at a flexible environment or when in motion and all that. Okay, please, lastly, someone is writing here, when preparing the target, we did cleaning of the target. You said that some ligands were used to stabilize the protein. If those ligands for stability get removed, does it not affect the structure of our target? And won't this affect the out? Okay. I swear. Yeah. Actually, like I mentioned, those co-crystallized ligands were intentional. Okay, they were intentionally there for stability purposes, just to enable the 3D elucidation of that particular target structure. So once the 3D structure is elucidated, their function or goal is almost achieved. Okay, so what do you do when you want to now study further with respect to just that target? You don't need those co-crystallized ligands again, except the original substrate of that particular target. In this case was naproxen, okay, which we could use to trace the ray catalytic triad or the ray catalytic subunit of that particular target. If you had visualized this protein and the naproxen in its active site, you discover that this arginine 120 I mentioned about is actually attaching itself to the ligand incoming. Try to tell you that that amino acid is actually the one responsible for attaching to incoming ligand and as well initiating electron abstraction through the neighboring subunit and all that, which when you now target it during site-specific molecular docking, you are as good as interrupting the rest activities of the protein. So the ligands actually plays no other role after that, and then they won't affect the outcome of your docking. Thank you. Okay, I think that is all. Thank you so much for your time.
Thank you so much, Samson. That was a wonderful presentation. Right now, we'll be going over to our lunch break. So we have snacks for those on site. So if you're on site, just stay back. We have some good snacks for you. While the snacks are being shared, please listen to our facilitators on site. They have a few announcements to make. And then after that, we're going to come back for the second half of our program, which starts at 1:30 p.m. So if you go out for the lunch break, please make sure to come back and join us. Thank you so much. Thank you, Samson. That was a wonderful presentation. So over to you, Chisom. You can take over from me now.
Good afternoon, everyone. Hope you're having a good time. Good afternoon, everyone. I can't seem to hear you. Good afternoon, everybody. My name is Umike Ikenna Emmanuel. Please, can you turn to your neighbor and tell him your name so that we'll not be strangers again? Hello? Are we having a good time? Officially, I'm sorry for the breakdowns and, the interruptions that happened at the beginning of the event. It wasn't scheduled to happen, but, let's say we are in Nigeria and some things don't go out the way it's supposed to be. So officially, I'm here on behalf of AODI. I welcome you to 2024 AODI Science Lecture / Workshop. This workshop held sometime last year, and it was a great event. This time, it's still a great event since you are enjoying and since you are learning and since you're asking questions. So I believe it's going well. I believe my facilitators, online can see and, testify that yes, it's going properly. So I'm just here to talk about a few things, which is the AODI, Africa of Our Dream Education Initiative. I'm wearing that, it's an organization, an NGO set up for nothing else than, the English is all about. Africa of Our Dream Education Initiative. And what's it all about? We are trying to commit education to us. Most of us in Africa or in Nigeria, as we know, we'll be saying most things about education. We are saying school has come, Abi. Last, I shouldn't have been here. I shouldn't have come here. I should have been learning Forex and doing other stuff, doing other business, right? Most of us are slipping out of education. We are going back to, we are sleeping, right? So education is not what it used to be before. They will tell you before, maybe my dean or the HODs will tell you back in our days, we did this, back in our days, we did that. Nowadays, if you go for practicals and stuff, they will tell you, imagine this, imagine that. It's no longer the way it used to be, right? So we are trying to bring back that dream. We're trying to bring back that, what education is like, what education is like abroad. When you go abroad, right? That's why most of us want to japa, want to leave this country and go abroad, because back there we have equipment, we have stuff that, make life easier for us. You can synthesize your compounds, you can do many things that you want to do and actually learn and actually, maybe win a Nobel Prize. But in Nigeria, you can't do that. So the organization was set up for that, to achieve the dream back. Let's education flourish in Nigeria or in Africa, like the name implies. So I am a campus ambassador. I have been representing this organization for some time now, since they came here, and it's been wonderful. Most of the things you see here organized have been done by campus ambassadors, since our AODI team, due to one thing or the other, they are not here with us today. So you see that it's a wonderful program. I have been trained. Most of my campus ambassadors wearing the t-shirts you see, they have been trained to work on something like this. And that's why I'm here to talk to the students, probably the ones in first years and second years, people who are still students, that you can join this program. It's that out there, when you go abroad, the main thing you're supposed to volunteer program is part of what they look out there for. What have you done? Right? So that's what it's about. You join the program, you get trained. If there are events like this, you go for events like this and represent the organization and preach education, preach positivity. That's Africa. Tomorrow, we are believing that we can grow tomorrow. Those stuff that we are running away from Nigeria to go abroad to do, we can actually do them right here in Africa and make Africa grow and become, a great continent. Thank you very much.
Please, a round of applause for Ikenna, please. Okay, good afternoon, everyone. My name is Chisom, and I think Ikenna has really said everything there is to say about the Campus Ambassador Program here at UNN. But I just wanted to reiterate the fact that we actually started this program at AODI because we wanted to be closer to the students. So there are a lot of programs that you can find for the university, and then those organizations are closer to the university, and then they are talking to the vice chancellor, the admin, and all of that. But we wanted to have a program that would make us actually closer to students. We want to hear directly from the students. We want to know what they need exactly. We want to see where their issues are and then treat those issues directly. So I want you to understand that this particular program is exactly made for students. So it was flagship here in UNN, but then we have our students at OAU, UniJOS, I think across 4 campuses right now. So it's an integrated movement where we want to basically serve the Nigerian students. The program is for you. And here at UNN, I think they're going to be making a call for volunteers to join the Campus Ambassador Program. If this is what you're interested in, helping other students get these kind of programs across to them, listening into forums where other students actually are, listening in and seeing what are your key needs as students, then bringing this information back to us while we seek funding, while we seek for ways to make sure that we get programs such as this to actually bring to you guys. If you want to join, you only need to just speak to Ikenna or any other campus ambassador that you see around you. It's a program that is actually designed for you. Just like Ikenna was mentioning, it's going to groom you in a lot of ways. I don't think it would be surprising if I told you guys that the bulk of what we have here right now, what we actually pulled through today, was actually initiated by the campus ambassadors at UNN that we currently have. Almost everything that you're seeing right now set up was actually done by these guys. So it helps you, it grooms you. You work with teams. You are a team lead, a team player. You know certain things about hosting events and all of that, and you can even actually help impact the life of other students around you. So generally, this is a wonderful opportunity for anybody who is a student, who wants to give back to the society, and who also wants visibility, to be seen as a thought leader. You want your thoughts to be heard. You want the things that you actually want to happen in your university heard. Just like Ikenna was saying earlier, there's a lot of things that and are not happening at our campuses, but you can be the voice. You can be the person who champions whatever it is, a women's rights movement, for whatever it is, whatever project you want. You bring it to us, we look at it, we calculate, and at the end of the day, we have things for all our students. Thank you very much. And remember that if you need to join the Campus Ambassadors program, just speak to Ikenna or any campus ambassador that you can find, and then you register. There will be a process where you're screened, and once you meet our feet, that's it. That's all it takes. Thank you very much.
Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined. Artlist.io. Music licensing reimagined.
Hello, everyone. Hope you're having a good time. You're not. What do you want?
Artlist.io.
You want small meat pie? Go and buy. Who wants more drink? Nobody wants more
Music
drink. Again.
licensing reimagined. Artlist.io. Music 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 licensing reimagined. Artlist.io.